to increase the food production for our country, 3 methods are used actually. First, one is agrochemical based type of agriculture. Agrochemical. Chemical means what? Yes, chemical pesticides, chemical fertilizers, they will use all those. So that is the first method, agrochemical based. Second, they can be organic based, fully organic. What's the meaning of organic? They won't use chemicals. As fertilizer and all, they use what then? They are not using agrochemicals, means what they use? Very good. Cow dung. Like that, forms are called organic. Or they will use fertilizer, it will be biofertilizer. Like some of the algae, they can be made as dried and made as biofertilizer. So they will give nitrogen, phosphorus, sulfur and all they will give. So that kind of agriculture is called organic. Organic agriculture. Now these both they can use with improved variety of crops like hybrid seeds, that and all they can use. But to make a hybrid is difficult. Make a hybrid difficult means sometimes a hybrid we are looking for what qualities that will not express in the hybrid. Because genes will separate, it will not come into the product. Like suppose you want a pink rose, you are pollinating red rose with white rose. We are expecting to become, in the F1 generation, we are expecting we will get pink. But sometimes due to some genetic problems, pink will not come. Maybe only red will come. Can be due to law of dominance. So genes we cannot predict how they behave. So that's why the last one is genetic based, where we are actually injecting the gene into the embryo. Then definitely that gene will get expressed. So instead of going for traditional type of agriculture, we can get genetic based. That is the last method, genetically engineered crop. Like if you want pink roses, they will find out which gene coded for pink color. They will find which gene it is. Then that gene they will inject into the seeds or the embryo of that flower or the fruits of that flower. So what will happen? They have to express that genes code for pink color. So the rose will be pink itself. Whole field there will be pink roses. So that we can trust more than using, depending upon the pollination. Pollination might not work. Traditional one might not work, so we can use this method. So these are the 3 methods written here. Though green revolution, now what is green revolution? Green revolution was brought in order to increase the plant yield. And it increased also. At first it increased. Population is high, that's why they used green revolution techniques, which includes mechanized tractor, hybrid seeds, high yielding variety, all they used. But there were problems. What was the problem? The fertilizer which they used was chemical. So when you use chemical fertilizers, chemical pesticides, it will actually spoil the quality of soil. Then future generation, that crops will not grow there. That is one problem. Next is hybrid seeds. Every time buying hybrid seeds is costly also. So that's why green revolution is not so good. Even though initially it helped. So they have written one word here, GMO. GMO means genetically modified organisms. Genetically modified organisms means what? Genetically modified means genetically they are changed for some purpose. For example, we have a hormone, insulin. You know what is the purpose of insulin? To reduce sugar. Whatever food you are eating, from there the starch has to be converted to sugar, and the sugar has to be used by our cells so that it can give you energy. For that insulin is required. But due to some reason, suppose that hormone is not produced by the pancreas, there is some problem with your body, became sick, so pancreas stopped producing insulin. Then what will happen to whatever you eat? That sugar. Sugar will remain in the blood itself without going into the body's cells. And if the cells are not getting glucose, cells will not be active. They will be deprived of energy. So that's why insulin is needed to be produced in bulk and injected to the person, or they can have it as tablets. So what they did is, they found out which gene is coding for insulin hormone. From human beings they found out. Genetic engineering, DNA, base sequence and all, with that they found out which gene codes for this hormone. And that gene they injected into bacteria, like E. coli. They put into where the bacteria is growing, in that culture medium they put this gene. So they took this gene inside, that gene went and attached with the bacteria's gene. But here the bacteria got modified. Modified means changed. First that E. coli was not producing any insulin. Now their gene got injected with our gene. Human insulin gene is there inside its body now. So what will happen? That bacteria will start producing. Now this suppose bacteria's genes. Now our gene is inserted into it. Our gene is there. This is human gene. Human insulin gene. So what happened? As this bacteria is growing in the culture medium, bacteria will start producing and expressing this gene, and it will start producing hormone, insulin. Later, after purifying, processing and all, these insulin hormones can be taken off and they can be made into tablets or syringes. They can be distributed. So the advantage is that the bacteria here is only one, but the bacteria can divide. So this will get divided into form 2. The 2 can again get divided to form 4. That means the amount of insulin produced will be high if you culture this bacteria. So this bacteria is genetically modified. Why? Because first this bacteria was not producing insulin. We did something. We inserted the gene. That is why it is producing insulin now. So it's called genetically modified organism. One example only I told. Like there are many organisms they genetically modified. One is our vaccine also. Hepatitis B vaccine. There are biotechnology companies there, like Hyderabad, Bangalore and all, like Biotech. They all do this genetic engineering and they create vaccines in bulk. That vaccine will be distributed and then we will stay immune. What are the GMO advantages? Genetically modified organisms. First is plants. They make the plants tolerant to abiotic stresses. They make the crop stress tolerant. For example, cold, drought condition where there is no rain, drought. More salt in the soil, heat. So they have genetically engineered some crops and those crops, they are very strong. So even if there is no rain and all, they can withstand that. Still they will be able to grow. So like that, genetically engineered crops can be made. Second, they will be having less dependence on chemical pesticides. Now, for example, morning I told you Bt Cotton. Now there is no need to apply. If you are already making the genetically engineered cotton plant, there is no need to kill the pest using chemicals, because from within the genes are expressing. So that is the meaning of the second point in this. Reduce reliance. Reduce reliance means the dependence. Reduce dependence on chemical pesticides. This you can give example. Bt Cotton. Bt Cotton is genetically engineered. They have put inside Bacillus thuringiensis 4 genes. Genes are called CRY, okay, CRY. That genes they have put inside. So as the plant is growing, they start expressing these genes. Toxin will be produced. So no pest will attack that plant. Like that, other importance, help to reduce post-harvest losses, increase mineral usage, enhance nutrition value of food. Reduce post-harvest loss. Minerals used better ways. Minerals used efficiently without any wastage. They have injected some genes which can use the minerals properly from the soil. Usually when we are putting fertilizer, chemical fertilizer, plants, they have a limit to take it. Sometimes they will not absorb all the fertilizers properly. At last it will rain and then these fertilizers will leak into the surrounding water bodies, causing water pollution. And the plants are not good enough also. So instead of applying the chemical fertilizer, if you are genetically modifying the plant, they will be able to use minerals properly from the soil. So there will be no wastage. And what's the meaning of post-harvest loss? After harvest, sometimes some fruits are there, they become ripe very fast. Fast they become ripe. Before they have been transported to sell in the market, they will become ripe. Overripe fruits, vegetables, people won't like to buy. So genetically they have been modified such a way that they will become ripe slowly only. I just gave you example. Okay. So in that way, post-harvest loss can be stopped. Nutritional efficiency. Increase nutrition value of food. For example, golden rice. Now this, how the question can be asked? Golden rice, how is it different from our normal rice? What is special about golden rice? Options are there because it is having gold color, gold plating. It has vitamin B, it has vitamin A. What is the answer? Yes, that is the answer. So golden rice has what? Vitamin A. Now this is a cotton plant, okay? Cotton plant, nicely fluffy white cotton is supposed to come. But there are some insects what they have given as work. They have given here the heading Lepidopterans. Lepidoptera, these worms. Even butterfly also comes in this order, Lepidoptera. So tobacco budworm, armyworm, those worms have the habit of eating the leaves. They eat the leaves. They also cause rotting of the cotton. So this causes lots of losses for the farmer who is cultivating it. And some of the bugs and beetles are also there. They belong in the order Coleoptera. So that's why they have written there Coleopterans. Coleopterans means some beetles. Some beetles and some flies are also there. Now Coleopterans, beetles will come in. Dipterans means it belongs to Diptera order. That flies will come. Fly, some mosquitoes. They all harm the plant, but based on what is harming the plant only, they create genetic engineering in the plant. So now here, this is one of the worms. That worm is eating and enjoying. But what will happen? All the leaves because bad means then this flower and all will not come. So there will be loss for the farmers. So what they do is they will get in sachet. Sachet means packet. Like shampoo sachets are there, like that. They get in sachets spores. Spores of which bacteria? Bacillus thuringiensis. Bacillus, the word bacillus comes when the bacteria is rod shape. Okay. Bacillus thuringiensis. That is its species name. This one spores are chemically they are available in the market. So they will take from this and they will spray it. So when that is sprayed, while the worm is trying to eat the leaves, by mistake it will swallow the spores. And once the spores reaches into its intestine, there is what medium in the intestine? Yes, very good. They have alkaline medium inside. Alkaline gut it is. Gut means intestine. Then the spores which is there, they will become active. In the gut spores where they know they will become active. Once they become active, they are toxic, so they will cause holes in their intestine. And if the intestine is having holes means then they cannot survive. They will die. So this is what happens. They will die, but it's good for the farmers. There's nobody now to eat. Leaves. Plant will stay healthy. They can even harvest the cotton balls. They can send it. So this is the method. How they kill these worms of these category and insects also. Now each time buying and spraying is difficult. That's why genetically these genes were injected. They got injected into the plant when the plant was embryo. And that gene's name is, this gene is coding for, this gene codes for Bt toxins. This gene codes for Bt toxin. And that gene is called cry. The gene is called as cry gene. That will be injected into the plant. So the plant from within, they will keep expressing this poison. So no insect can attack it. They will be resistant. So we don't have to apply this thing. Pesticide, chemical pesticide, no need to apply. Because from inside itself it is resistant to all the insects and worms. Now this gene, different gene, Try2AB. It is injected to kill cotton bollworm. Try1AB. It is given to control pondworm. Suppose this is a tobacco plant. There is a worm that is infecting the roots of these tobacco plants. This worm's name is Meloidigyne incognitia. Incognitia. In their body, they have one kind of RNA and you translation. Yes. So what happens in Meloidigyne incognitia is there some special RNAs are there. That RNA is producing a protein that actually causes damage to the roots of tobacco plant. So what the attempt is to block that RNA from expressing. That RNA has to be blocked. For that, what they do is they are injecting one complementary RNA. That RNA they are getting it from some viruses. Some RNA containing virus whose nitrogen bases has complementary base pairing with Meloidigyne incognitia's RNA. Like it will also have an RNA, no? This one's RNA is there inside. Suppose their pattern is like this A, G, C. Suppose Meloidigyne incognition's RNA is like this. Only example, okay? And the virus's RNA is having its complementary base. So it will be, what will be the complementary base then? If it is AAGC, here what it will be? TT, yes. TT, CG. Yes. Like that RNA they will find out. And they will isolate it. After isolating, they will introduce this RNA. This RNA they will introduce into the plants. So either they can use injection method or another method which is written in the text is bacteria method. There is a bacteria who causes infection to the plants. That infection causing gene they will remove. Instead into the bacteria they will put this RNA. Now this RNA is called as antisense RNA. They call it as antisense RNA, and the RNA of the Meloidigyne they simply put it as sense RNA. So this RNA they will introduce into the plant. With a bacteria. Bacteria's name is Agrobacterium polyphasians. First they will culture this RNA in where this agrobacterium is growing. Then the bacteria will get it inside the body. That bacteria they will allow to infect. So when this bacteria is infecting the plant, instead of causing infection, they will introduce this RNA into the plant. So this RNA will get inside the plant. Antisense RNA will get inside the plant. When it entered into the plant, it saw the mRNA of Merodegium incognita here in the roots. So that was having the complementary base, this one, no? AAGC. So what will happen then? One is Merodegium incognita's RNA, that is called sense RNA. That RNA is there because Merodegium worm bit it. It infected it. That's why into the plants that RNA came, which is called sense RNA. What did we introduce? We introduced a complementary RNA, which is called antisense RNA. Since they both are complementary, they will pair. Antisense RNA, sense RNA, they will pair. And RNA will become double stranded. Now here, what will happen then? It is getting blocked. Which RNA got blocked? Which RNA got blocked? Sense RNA got blocked. That is, Merodegium incognita's RNA got blocked. Who blocked it? Yes, antisense RNA, which they got it from some virus. It had complementary base. That one interfered with the RNA of worm. That's why worm cannot destroy now. Worm can bite, but even after biting also, it cannot destroy this plant. Because the plant has what as a protection? Plant has antisense RNA. So there will be pairing between antisense RNA and sense RNA, and because of this pairing, the mRNA of Merodegium got silenced. When do we say this method of silencing is also called RNA interference? Usually questions are asked for like this topic, RNA interference.